Now that we've looked at the properties of amino acids, we're ready to discuss the different types of bonds that contribute to protein folding. As we saw, amino acids are linked together by peptide bonds, and this is a type of covalent bond. Other types of bonds, which are mostly non-covalent bonds and interactions, are involved in protein folding. And these include hydrogen bonds, ionic bonds, van der Waals and hydrophobic interactions, and disulfide bonds. Of these, only the disulfide bonds are covalent. Now, most of these interactions are occurring between the R groups of the amino acid residues. However, as you'll see later, there are some important interactions that occur between atoms of the polypeptide backbone. Recall that denaturation experiments have shown that the information required for protein folding is contained within the polypeptide sequence. And this is because protein folding is determined by the chemical properties of the R groups of amino acid residues. And the chemical properties of these amino acid residues dictate the type of interactions and bonds that they're able to participate in. So a protein will acquire its most thermodynamically favorable conformation based on its amino acid sequence. And by the way, the word conformation means shape. Of the types of bonds involved in protein folding, the only covalent one is the disulfide bond. This bond forms through an oxidation reaction between the sulfurs of two sulfohydryl groups on cysteine residues. Since this is an oxidation, it involves the loss of electrons. And these electrons are lost as two hydrogens, so both the protons and the electrons are lost, as is often the case in cellular oxidations. Since these bonds form through oxidation, they can be broken through reduction. The reduction and oxidation of disulfide bonds is actually the basis for the hair treatment known as the permanent or the perm, where the hair is artificially curled. Hair is composed of keratin, which is a fibrous protein, and there are different types of keratins which are found in different tissues. Keratin is composed of two helices, as you can see here, that are wrapped around each other, and this is known as a coiled coil. The strength of the keratin comes from disulfide bridges or disulfide bonds, which cross-link the coils together. Now, the different types of keratin found in the body differ in the number of cysteine residues and thus in the number of disulfide bonds. For example, the keratin in hair, which is flexible, has fewer disulfide bonds compared to the keratin in fingernails, which is rigid. To make the hair curly, it is first treated with a reducing agent to break the existing disulfide bonds. Then the hair is put into curlers and an oxidizing agent is added to create new disulfide bonds. The chemically induced curls are eventually diluted out by new hair that grows from the scalp and takes on the shape of keratin fibers that is specified by the amino acid sequence of the corresponding keratin genes. Disulfide bonds can be intermolecular, like the ones shown here, which are happening between cysteine residues on different polypeptides. But they can also be intramolecular, happening between cysteine residues of the same polypeptide. And these intramolecular disulfide bonds can bring together distant amino acids, and they can stabilize the three-dimensional conformation of proteins. The intermolecular bonds, which happen between different polypeptides, can serve to bring together these polypeptides in multimeric proteins. Another type of bond that is important for protein folding is the hydrogen bond, which is a non-covalent bond. The R groups of some amino acid residues can form hydrogen bonds because some can serve as hydrogen bond donors, while others have atoms that can serve as hydrogen bond acceptors. For example, the amino acids serine and threonine have hydroxyl groups. Hydroxyl groups have an oxygen that is covalently bonded to a hydrogen. And the hydrogen has a partial positive charge since it is bonded to an oxygen which is highly electronegative. This electropositive hydrogen can form a hydrogen bond with a second electronegative atom such that the hydrogen is essentially shared between the two electronegative atoms. Hydrogen bonds not only form between the R groups of two amino acids, but they can also form between the R groups of an amino acid residue and water, and between the atoms of the polypeptide backbone, as we'll see later. Hydrogen bonding between amino acid residues and water help to make proteins more soluble in water. Like disulfide bonds, hydrogen bonds can form between amino acids that are located far apart from each other in a polypeptide chain. And as a result of that, these will be brought closer together during protein folding. As you know, hydrogen bonds can also occur in other macromolecules like DNA, and they can be inter and intramolecular, both in proteins and also in other molecules. Now, even though these hydrogen bonds are weak bonds, when many of these bonds are present, their combined effect provides significant stability to macromolecules. Another type of non-covalent bond that is important for protein folding is the ionic bond, since the R groups of some amino acids are charged at neutral pH. Ionic bonds are attractions between oppositely charged ions. For example, an ionic bond can form between the negatively charged carboxyl group of the amino acid aspartate and the positively charged amino group of the amino acid lysine, as shown in this figure. Since charged amino acids are common within the active site of enzymes, electrostatic attractions like this one are important for the interactions between enzymes and their substrates. Recall that although ionic bonds are strong in vacuum, they are weakened in the presence of water because ions form weak ionic interactions with water, which essentially compete with the interactions between the oppositely charged ions. This happens because it is thermodynamically favorable, but the reasons for this are beyond the scope of this course. Although these bonds are weak in aqueous solution, their combined effect is strong and they can serve to stabilize interaction within molecules or between macromolecules, like the interaction between the negatively charged phosphate groups in DNA and the positively charged amino acid residues in histone proteins. The ionization state of amino acids is affected by changes in pH. At physiological pH, the carboxyl and amino groups on the backbone of an amino acid are ionized, as is the carboxyl group and amino groups on side chains. We're going to focus here on what happens to the side chain because the amino group and carboxyl group of the backbone lose these charges when they're incorporated into a polypeptide. Now, suppose that we alter the pH of a solution, which essentially means that we're changing the concentration of protons in the solution. An amino acid that has a carboxyl group as its side chain, like aspartate or glutamate, will become protonated at low pH. Now, this change in the ionization state of the carboxyl group will in turn affect the ionic bonds that can form between amino acid side chains, and that can potentially affect the interaction between substrates and their enzymes, for example. A similar situation would result if we increase the pH so that it's very high. Now you can see that the carboxyl group of the side chain is ionized at high pH, just like it is at physiological pH. However, for amino acids that have amino groups as their side chain, that amino group is ionized at physiological pH and it loses a proton, so it loses a charge at high pH. So because of this, we would have a similar effect whether we had decreased the pH a lot or raised the pH, since that change in pH would alter the charges in these groups, and by altering the charges in these groups, it would alter the types of bonds that they could engage in. So the take-home message is that changes in pH can alter protein structure by disrupting the ionization state of the side chains of amino acids and in turn affecting the formation of ionic bonds between these groups. Besides ionic bonds, which are based on interactions between oppositely charged ions, other interactions exist that are also based on charge but occur in molecules that are not actually charged, and these are known as van der Waals forces. Even a molecule that does not have an overall charge can have a transient or temporary uneven distribution of electrons, and that's known as a dipole. At very short distances, atoms show weak bonding interaction because of their fluctuating electrical charges. Now, these interactions are transient since the distribution of electron fluctuates. Although individual van der Waals interactions are weak, These can be significant in macromolecules that fit closely together, like an antibody and its antigen, because many atoms are involved. The last type of interaction that is important for protein folding are the hydrophobic interactions, and these are not bonds. Recall from the last lecture that water forces hydrophobic groups together. These interactions are not due to any intrinsic attraction between the nonpolar molecules. Rather, they result from the system achieving the greatest thermodynamic stability by minimizing the number of ordered water molecules that are required to surround the hydrophobic portions of the molecules. As we saw earlier, this hydrophobic effect results in amino acids that are hydrophobic being buried within the core of cytosolic proteins or in the transmembrane segments of transmembrane proteins. If a protein were composed of mostly hydrophobic amino acids, it would be insoluble in water. Although not shown here, water molecules would form cages around a protein like this. For this reason, proteins containing mostly hydrophobic amino acids tend to be localized within membranes. And even membrane proteins are amphipathic, since the loops that are located outside of the cell and within the cytosol are composed mostly of hydrophilic amino acids, whereas the transmembrane segments are composed mostly of hydrophobic amino acids. On the other hand, if a protein was composed only of hydrophilic amino acids, the protein would remain in a highly extended conformation, as this would maximize the interactions of hydrophilic amino acids with water. Because most proteins are amphipathic, containing both hydrophilic and hydrophobic amino acid residues, hydrophobic interactions play a central role in protein folding. So protein folding is a balance between the tendency of hydrophilic groups to interact with water and the tendency of hydrophobic groups to avoid interaction with water.